And this is Chapter 12, Part 7. We're going to look at graded potentials. A graded potential is a small deviation from the resting membrane potential that makes the membrane either more polarized, which makes the inside more negative, or less polarized, making the inside of the cell less negative. So polarized, we're going to make the inside of the cell more negative. So when the response of the membrane makes it more polarized, this is termed a hyperpolarizing graded potential. So as you can see here on this little graph, we start here at -70. Actually, I'll change the color here. We're going to start here at -70, and we're going to have a response in the cell that actually causes the membrane potential to go a little bit more negative, maybe about -75. So that's going to be hyperpolarizing or more polarizing. Now, on the other hand, when the response makes the membrane less polarized, this is going to be termed depolarizing. And depolarizing makes it less polarized. So we're starting here at -70 at resting membrane potential. And we're going to have a response in the cell that actually causes the cell to become less negative. So -60 is a little bit less negative on the inside of the cell than -70 is. -60 less than -70, and that's going to be depolarizing. A graded potential occurs when a stimulus causes mechanically gated or ligand-gated channels to open or close in an excitable cell's plasma membrane. Now, typically, the mechanical and ligand-gated ion channels are found in the dendrites of sensory neurons. So sensory neuron dendrites, we're going to find both the mechanical and ligand gated, these ions both there. And ligand gated channels are far more numerous in the dendrites and cell bodies of interneurons and motor neurons. Motor neurons. So we're going to have these in the dendrites and the cell bodies. We're going to have more ligand-gated channels there. So we have these, so we know that graded potentials occur mainly in the dendrites and the cell body of a neuron. So that's where we're going to have most of these graded potentials, the dendrites and the cell bodies of neurons. So here in this diagram here, we have pressure that's applied to a mechanically gated channel here. We've applied pressure. On the left, it's going to be closed, and then here we could see on the right it is open. We also have an acetylcholine ligand-gated channel here, the purple one. So you can see here when acetylcholine actually binds to the receptor, it opens and allows ions through the now open channel. And this last one here, we have a glycine ligand-gated channel. On the left picture, you can see it's actually closed, so we're not going to be able to get any ions through to the other side of the plasma membrane. But once these glycine actually bind, it's going to be open and allow ions through there. In this particular case is chlorine. So to say that the potentials are graded mean that they vary in amplitude. It's essentially what we're talking about when we talk about graded potentials. That they vary in amplitude. And this amplitude depends on the size of the stimulus. So these amplitudes of the potentials are actually larger or smaller depending on how many ligand-gated or mechanically gated channels have opened or closed, and also for how long each remains opened for. The opening and closing of these ion channels alters the flow of very specific ions across the membrane, and this produces a flow of current that's very localized, which means that it spreads to adjacent regions along the plasma membrane in either direction from the stimulus source for a short distance, and then it gradually dies out as the charges are lost across the membrane through leak channels. So this mode of travel by which graded potentials die out as they spread along the membrane, this is known as decremental conduction. I'll put this here. Decremental conduction. So that's the idea that if we have, say this is a dendrite here, if we have a graded potential, a dendrite on the cell body, just a little thing here. So if we have a graded potential that comes up right here, as we go further away from this point of stimulus, or this stimulus source right here, it's actually going to die out as we go further away. So this is decremental conduction. So because these graded potentials actually die out within a few millimeters, graded potentials are useful for very short distances. This diagram here shows how the stimulus strength is corresponding directly with the change in membrane potential. So this smallest stimulus would actually produce a very small change in the membrane potential, and then the stronger stimulus actually produces a larger change in the membrane potential. Although an individual graded potential undergoes this decremental conduction, it can actually become stronger and last longer by summating with other graded potentials or adding on other graded potentials to it. Summation, this word right here, this is the process by which graded potentials are actually added together. So if we have two depolarizing graded potentials, they summate, the net result is a larger depolarizing potential. And if two hyperpolarizing potentials summate, the net result is that we have a larger hyperpolarizing graded potential. And then if we have two equal but opposite graded potentials summating, we have one depolarizing and one hyperpolarizing, they'll actually cancel each other out. And the net result is that we would have a graded potential actually disappear. So in this diagram here, we have this first stimulus right here, actually going to cause this little graded potential, but as you can see, it very quickly starts to die off. And then we have this stimulus 2, that as it dies off, the second stimulus causes an increase in that graded potential, and causes it to stay around for longer before coming back down to the resting membrane potential. And that's going to be the end of chapter 12, part 7.